Well, thank you all for coming, and thanks to Julie and Joe specifically for the invitation to speak today. I'm Julie Ziobro. I'm a pediatric epileptologist and a basic scientist at the University of Michigan. I work in the lab with Jack Parent, who some of you may know. He spoke here a few years ago as well. And I'm going to talk today a little bit about some of our work on examining inhibitory interneurons in PCDH19-Clustering Epilepsy. I don't have any relevant disclosures. So a couple objectives for today. So I'm going to talk a little bit about the role of interneuron pathologies in the DEEs in general. And then I'm going to talk more about our specific rodent modeling of PCDH19 clustering epilepsy, some of which you've already heard from Dr. Umemori, so he set me up nicely, so thanks for that. And then we'll explore the role of interneuron pathologies in PCDH19 clustering epilepsy. Or I actually like Hisashi's PCDH19 related disorders because, as he mentioned, they're not all, not every patient has epilepsy. So maybe we need to recoin a new term, even though we just came up with this one a few years ago. So the developmental and epileptic encephalopathies are a group of disorders, and the definition by Ingrid Scheffer and the group in 2017 is that epilepsy is associated with developmental impairment that may be due both to the underlying etiology, so the developmental encephalopathy, and the superimposed epileptic encephalopathy, so the epileptic encephalopathy. So not necessarily that the seizures cause the cognitive disorders, but some of these are kind of go together. And when we think about the standard paradigm of epilepsy in general, and this is in a very kind of simplistic term of ions and synapses. And to develop epilepsy, you either have to have too much excitation or not enough inhibition. So too much of the gas or not enough of the brakes, right? So if there's too much excitation, we know that can be related to problems with sodium channels and excitatory synaptic activity. If there's not enough inhibition, this may be related to potassium channels, or it may be related to not enough of the inhibitory synaptic activity, including GABAergic synapses. So I'm going to focus a little bit more on this lack of inhibition. So we got a little bit of a development lesson from Dr. Umemori looking at the excitatory neurons, but I'm going to talk a little bit more about these inhibitory neurons. So inhibitory neurons are also known as interneurons, and they do this kind of wacky thing in development. So the interneurons are derived from these caudal ganglionic eminences. It's off. Okay, never mind. Oh, here. So it's called the medial ganglionic eminence or the caudal ganglionic eminence, the MGE or the CGE. And then what they do during development is the neurons are born here. And instead of migrating in this radial fashion, like the excitatory neurons, like Dr. Umemori had talked about, these inhibitory neurons actually make this long trek all the way from the eminences to wherever they're going to end up in the cortex or the hippocampus or wherever else they'll end up in the brain. These migrating neurons have to have a lot of extrinsic cues, so a lot of other factors that make them go where they're supposed to go. And we know from other disorders and other DEEs that defects in this migration of these cells can lead to abnormal excitatory and inhibitory balance. An example of this would be for a disorder of GABAergic interneurons called ARX. And this was studied by Eric Marsh in 2009. And you can see in his pictures, this is looking specifically at some inhibitory interneurons. And in their ARX model, there's a change in where these inhibitory interneurons are located in both the hippocampus and in the cortex. The term interneuronopathy was coined by Bill Dobbins in 2005, so kind of way back then. When he recognized that some of these interneurons were not going where they're supposed to go in some of these genetic epilepsies. So developmental interneuronopathies includes a subset of these disorders that result from abnormal embryonic proliferation, meaning when cells are being born, differentiation, meaning what types of cells they turn into, and then the migration, if they end up where they're supposed to go. Here's some of the other epilepsy syndromes that we know are related to interneurons. So like I talked about ARX, which causes an X-linked lissencephaly, so kind of a smooth brain syndrome, where you don't get the normal gyri and sulci. And we know that there's abnormalities in the interneurons in ARX. And then there's a couple other genetic syndromes, including DCX and PAF-AH1B, which is another lissencephaly syndrome. In addition, there's been some evidence that things like West syndrome, so infantile spasms, that can be caused by either structural changes or genetic changes, actually have some impaired interneurons as well. And then also in Dravet syndrome, which we talked about earlier today too, SCN1A mutations, there's a specific abnormalities in how those interneurons are firing, specifically in Dravet syndrome. Interneuronopathies have also been implicated for other forms of autism spectrum disorder and for schizophrenia as well. But are there others? Like, should we be looking more at the interneurons in all the DEEs, or is this very specific subset of disorders that are related to interneurons? So our question is, if there's other DEs that can be considered interneuronopathies, then can we maybe use this as a target for treatment for all of the interneuronopathies? So in our case, we wanted to look at this more in PCDH19. So as we reviewed already with Dr. Umemori, you guys know a lot about PCDH19-Clustering Epilepsy, and we know that it's characterized by these very varying phenotypes of early onset epilepsy, intellectual disabilities, and neuropsychiatric features. And I think the more we're learning about PCDH19, the wider the phenotypic spectrum is becoming. We know that the patients also often have these clusters of seizures that are provoked by fevers. They can be focal or they can be generalized. And the age of onset is quite early. Those who have an earlier age of onset tend to have worse intellectual function, though none of this is 100%. Over time, seizures often will decrease with age. And then, like we talked about earlier, there's a very unique inheritance pattern in which heterozygous females are affected, but not the hemizygous males, though we know that there's rare mosaic males who are affected as well. And this is kind of similar to what Dr. Umemori reviewed, that PCDH19 is a member of the delta cadherin subfamily. And we know it actually probably has more functions than just synapse formation. It probably also will contribute to neuronal migration, some circuit formation, and then the synaptic plasticity and maintenance. And as he mentioned, the exon 1 is really where most of the variants have been found, and they're including some loss of function variants or entire exon deletions, which would really suggest that the variants are probably all related to a loss of function. So you can have loss of function or gain of function when you have a genetic change. So the fact that having a whole exon deletion leads to the syndromes indicates that it's probably a loss of function mutation. And like we talked about earlier, there's no clear genotype-phenotype correlation, meaning that if you have the same change as another patient, it doesn't mean that they will have the same type of epilepsy or have the same outcomes. And I think some of that comes down to this thing we had discussed in the discussion, that there really might be a very location-dependent factor to the phenotype of the patient. A little bit like real estate, location, location, location may be incredibly important in this disease. There's been lots of work done along the way trying to look at what PCDH19 may be involved in. And importantly for our aspect, for this talk today, we're looking more at cell migration and neural circuit formation, and also at the GABAergic transmission. And then this is just reviewing what you guys have already heard, that in our PCDH positive cells only, there's normal cell-to-cell interaction and patients are symptomatic. In males, when you have PCDH19 mutations in all their cells, they're asymptomatic. But because of this random exon activation that happens in females, you get this mosaic pattern of cells in which the PCDH19 negative and PCDH19 positive cells coexist, but they don't interact normally. So our hypothesis is that PCDH19 is important for cortical interneuron migration, but that an unbalanced expression leads to the aberrant inhibitory synaptogenesis and may lead to the phenotypes seen with DEEs. So in order to examine this a little bit more, we had to actually first know if PCDH19 was expressed in those inhibitory interneurons. And some of this work has already been done, which is fantastic for me, but we know that PCDH19 expression comes on pretty early in development. So in the human, this is a 20-week human brain, and you can see PCDH19 expression is pretty high in the hippocampus and then also in the cortex. And then this is a P2 mouse, so a second day of life mouse. And there, as Dr. Umemori had mentioned, that there is expression in the cortex and also in the hippocampus. This expression seems to peak pretty early after the birth of a mouse. So around P7 seems to be the highest level of PCDH19 expression. And then it kind of slowly decreases after that. But this age in mouse development is really important for those interneurons during interneuron migration and for cell development in general. A few years ago, this group published some really fantastic data looking at interneurons in PCDH19. And they used a couple different techniques to look at how much PCDH19 is present in different areas of the brain. So this is in situ hybridization, so looking specifically at the mRNA. And they did find that there is PCDH19 mRNA present in the neonatal cortex, but then also in the ganglionic eminences as well. And they looked as early as E13. And they showed kind of the highest expression there at E16.5, so a couple days later, which is equivalent to several weeks in the life of a human. The expression changes a little bit, and you can see more in the what's called the limbic system as well. PCDH19 expression, it really changes over the mouse brain development, which means that it probably is very involved in the synaptogenesis of when those cells kind of make those connections, and mostly in the early prenatal and postnatal stages. And then we know that it's present in the precursors of the cortical inhibitory neurons, so the cells that are born in the MGE and the CGE. Additionally, this group did a little more work looking at the function of PCDH19 in those medial ganglionic eminences. So this was a pretty cool study they did where they actually took explants. So they cut out the area of the MGE and they grew them in culture. And then they were able to take an RNA that they could electroporate, so kind of incorporate it into the cells in the MGE. And when it incorporated, it expressed this TD tomato, so the red dye there. And they were able to look at how those cells migrated then from this MGE explant in the culture. And they found that in the control, the cells that had just a regular TD tomato, they didn't have PCDH19 knocked out, they made what we would consider kind of the normal migratory patterns where they really wanted to get out of the MGE and try and migrate into the different areas of the brain, though in this case it was just in a dish. However, when they knocked out PCDH19 in those cells, so these are the red labeled cells here, more of them stayed in the explant and they didn't migrate normally. Not as many of them migrated out of that explant, which would suggest that PCDH19 might also be playing a role in migration from the MGE, affecting how those cortical interneurons make it to where they're supposed to be. Other models that have looked at the inhibitory interneurons in PCDH19 include this recent paper from Ann Heduri's group in Boston, and she has a fish model. So a zebrafish model of PCDH19. And she found that having heterozygous expression, so similar to what is seen in females, led to a decrease in the inhibitory neurons that made their way to the tectum. So the tectum is kind of the main part of the fish brain. So they were able to show that there was a decrease in the number of inhibitory neurons in their heterozygous mice, but not in their complete knockouts. So we wanted to look at this more in mice. And like we've talked about a little bit earlier, and there'll probably be more discussion today with Dr. Thomas as well, is that rodent modeling of PCE has been really challenging for a lot of reasons. One thing is that mice are not humans. So mice often don't have the same phenotypes that humans have as much as we will look for them. Sometimes it's hard to interpret. Rodent modeling can be very background strain dependent. So if you get one mouse from Jackson lab and you get another mouse from another laboratory, they may have some different background genetics, which is similar to humans as well, right? Every person is not the same in their genetic background, but it makes it a little bit challenging when you're trying to have a very consistent model. And the other issues that we think there may be a lot of inconsistencies because of that random exon activation, because it is in fact so random. The pros of using rodent models is that it has really helped us to identify potential mechanisms, as you saw earlier. It's very exciting work to try and figure out exactly what PCDH19 is doing. And the behavioral phenotypes have been relatively subtle. We've tried looking for lots of other behavioral phenotypes that would coincide with what we see in the humans, and they tend to be very subtle or not statistically significant. There have been several groups that have showed lower seizure thresholds in several models, but again, this is not always a consistent phenotype in our mice as well. The cons, whenever you're trying to model something, it would be really nice to have a very robust phenotype so that when you manipulate it, you can see if there's a change. And we are still lacking having a very robust phenotype. We have not observed spontaneous seizures. Trust me, we've looked, I've EEG'd many, many mice. And then antibody labeling is challenging. So the antibody for PCDH19 is not always working well. So being able to identify where PCDH19 is expressed in the rodent brain can be a little bit difficult. So because of some of these challenges, we have used a transgenic mouse approach, which Dr. Mori very graciously has donated his mouse to me to work on some of these as well. And we've used a similar approach that he showed you earlier, wherein we take a PCDH19 knockout female, so she has no PCDH19 expressed, and we cross her with a male mouse that expresses a green fluorescent protein on his 1X chromosome. So when those mice have offspring, the females will have a green fluorescent protein on their PCDH19 normal allele, and they will have no green fluorescent protein on their PCDH19 knockout allele. The males will not express any GFP. So when we look at them, even early on, these are little mouse pups, and you can look at them with a fluorescent light and they glow. So I can always tell which ones are males and which ones are females even before you can really look and see what's a male and a female. We can even look in the embryos and know which ones are male and female as well. And similar to what has been shown by other groups, we can see that this PCDH19 mosaic expression is really leading to this unique segregation pattern very early in development. So we see it pretty early in an E12.5. So mice are born around 21 days. So this is about a week and a half before they're born. We can see that there's these clustering of the GFP positive cells, which would be the PCDH19 positive cells, or what we think is because it's a surrogate marker. And then the PCDH19 negative cells. So you see these really nice striping pattern. And in my very bad embryology at E10.5, you can see it even in the cortex that early in development as well. This cell segregation really continues through adulthood. So here in the E15.5, we can see this really nice striping pattern. When you compare it to a wild type female, so instead of taking a PCDH19 knockout female, if we cross a wild type female with that GFP mouse, you can see this very kind of random pattern of GFP and none of this cell segregation that you see in the PCE model. In the adult mouse, so P45, which is kind of a young adult, you can still see the striping pattern pretty explicitly in the cortex, and then also really strikingly in the CA1 region of the hippocampus. So because we saw it so strikingly in the CA1 region, it kind of led us to wanting to study that a little bit more. And again, this is just kind of another picture comparing the wild type where you see a very random pattern of the GFP positive cells, where in the PCDH19 heterozygous female, there's these stripes here that you really don't see any of the GFP positive, and the cells very much cluster together. So then the question, of course, is do these animals seize? And as I told you already, we don't see spontaneous seizures in them. So we've tried several different methods to try and induce seizures to see if they have a lower seizure threshold. There's a previous model that used chlorophyll, which is a chemoconvulsant, so a chemical that will cause seizures. And they did show some decreased seizure threshold. But we wanted something that was a little bit more applicable to the human condition. So we know that patients with PCE will often have seizures associated with fever. So there's this hyperthermia model in which you heat up the mice, kind of simulating a fever to see if they have a lower seizure threshold. Importantly, this really only works in their juvenile phase. If they were much older, they were more resistant to seizures. So we really settled on this P15 to 16, which is kind of a mouse equivalent of childhood. So they're relatively young when this model works. So just a short video of a mouse having a seizure. So this mouse is heated up and you can see him kind of at 42.5 degrees. He has a convulsive seizure. And then he's a little postictal for a while, and then they do bounce back. Oops, sorry. So we can measure this on what's called the Racine scale, which is a measure of how severe a seizure is when a mouse has a seizure. So a measurement of 2 is like some subtle head twitching, and even in wild type animals, they'll get the subtle head twitching when you heat them up enough. And measure Racine 3 is where they'll have like a little bit of rearing of one paw. And then it goes all the way up to Racine scale 6. And the Racine 6 seizure includes a prolonged tonic seizure that leads to death. 4 and 5 are both convulsive seizures. So you see in our model, the PCE mice and the male knockouts are the only ones who have convulsive seizures, whereas the wild type females did not have any convulsive seizures. And they were more severe in the PCE mice up to a racine level 6, where we actually had several mice die. We can also look at the temperature for the first seizure, and we found that there's a lower seizure threshold for the PCE mice compared to the wild type controls or the knockout males. Meaning that they'll have their first head jerks at a much lower temperature than the males, suggesting that they are more sensitive to hyperthermia. So we really wanted to know if this pattern of random X inactivation actually altered their seizure susceptibility. So again, like we talked about, location, location, location. This is one mouse, this is the right side, this is the left side of the hippocampus, and you can see very different patterns of GFP striping within even the single mouse. And then if you take this across multiple mice, you can see very different patterns from one mouse to another. So we were wondering if there was any correlation between how much striping they had in the hippocampus and how likely they were to have seizures when exposed to hyperthermia. And unfortunately, this did not reach any statistical significance, though we do think there may be a little bit of a signal there. So at this point, we don't see any actual stripe correlation with seizures. If we count the number of stripes, and we actually now have a computer program that will automatically do it for us to try and take away any bias that we may have when looking at these pictures. But when we try and correlate the number of stripes they have and the severity of their seizures, we haven't seen any significance. But notably, we're going to keep looking because we keep doing more of these assays. We have seen some abnormal electrophysiology in looking for markers of hyperexcitability in these mice as well. So this is in collaboration with Lori Isom's lab. They've done some patch clamping where you will actually take a slice of the brain, you'll stick an electrode into a neuron, and then just watch as the cell fires. And you can inject current into it to make it more likely to fire, or you can just watch it and see what it does on its own. And in these, in our PCDH19 mice, at P16, which is when we're doing our seizure assays, they have this abnormal pattern where they'll show different bursts and clusters of bursts, which is interpreted as them being more hyperexcitable, and it's a relatively abnormal pattern. So we do think there's probably some intrinsic factors within the cells that makes them a little bit more likely to be prone to seizures as well. And then kind of similar to what Dr. Umori has done, we wanted to know if these PC mice display any other abnormal behaviors. So there's a lot of different behavioral assays you can do in mice that are supposed to be kind of surrogates for things like anxiety or autistic traits or things like that. So in order to do this, we sent a bunch of mice to Scripps Research Institute, where they have a lot of experience doing behavior testing. And we sent mice who are 3 to 5 months old, and they went through a battery of 9 different tests that looked for things, testing for spatial learning, hippocampal function, anxiety, learning and memory, and social interaction. And unfortunately, we found differences by sex, meaning that males and females were different from one another, but there was no differences by genotype. And so when looking at the data a little bit more closely, when we look at some of these tests that were related to anxiety, so mice don't like when there's weird things in their cage. So if you put a bunch of marbles in there, the more anxious mice will be more likely. And I always put it in quote because we don't know if the mice are anxious, right? They can't tell us what they're feeling. But this is supposed to be a marker of anxiety. The more anxious they are, the more marbles they will bury. And it's kind of repetitive behaviors as well. And we saw that the knockout males that were actually kind of more chill, like they were less likely to bury these marbles, which actually kind of goes the opposite way that we would expect as well. You can also look at how fast they are in doing it. And we really saw no differences across groups. This is the elevated plus maze test. So mice are kind of dropped into a maze where part of it is closed and part of it is open. The more time they spend in the open, the less anxious they're supposed to be. So in our case, our heterozygous females were similar to the wild type females, and they spent a decent amount of time in the open. And then all the males kind of seem to spend more time in the closed arm. So again, really nothing interpretable or no real differences here that we can hang our hat on to say that there's a good phenotype. When we looked at a couple different other learning and memory tests that Dr. Umori had done, we looked at something called novel object recognition, and then we also used a water maze where they had to figure out where the target was in water, and they all really kind of performed similarly. I think what's interesting about some of this data is that there's a much bigger spread in the heterozygous females, suggesting that there may be some correlation with like the variability that we see in random axon activation as well. And that is something we're going to be looking at a little bit more. In tests of social interaction, they all essentially performed about the same, except for the knockout males who actually seem to be a little more sociable, which again, this is not very significant. So getting back to the interneurons though, and if the interneurons played any role in their seizures, we have done a couple different things looking at the types of interneurons that are present in these mice. So we were staining for multiple different interneurons and looking specifically at the CA1 region of the hippocampus. Since we do see such a significant segregation pattern there, we really wanted to focus in on CA1. And in looking at multiple different interneurons that are present in the hippocampus, we really found a significant difference in the parvalbumin cells. So parvalbumin positive cells are the most common of the inhibitory interneurons. And we do see a decrease in the number of parvalbumin positive interneurons in the PCE mice compared to their wild type female controls. The same was not true for cells that were positive for GAD67 or somatostatin. So we wanted to focus on this a little bit more in a slightly easier method as well. So we utilized some more transgenic mice and we crossed in a marker for PV positive interneurons. So we crossed a PV Cree mouse. So a mouse that expresses Cree on just the parvalbumin positive interneurons. And we crossed it with this reporter mouse, which expresses a TD tomato when it comes in contact with the Cree. So the offspring will express the TD tomato, so the red protein, on all of their parvalbumin positive interneurons. And then the green and not green are similar to as they were before. And this allows us a couple of things. So it lets us examine the parvalbumin positive interneurons at a very early age. It also lets us examine them in living tissue, so we can use those patch clamping techniques and specifically look at the activity of those interneurons, since we can identify them in living tissue as well. And it also makes our histology much more consistent because we don't have to rely on antibodies to actually show us where those interneurons are. So we've just started doing this not too long ago, and we have some data that at least is a little bit consistent with what we were doing before. So I have a technician in the lab right now who's fantastic with computers, and she's done a fantastic job of making sure that we can do this in a really non-biased fashion. So she has written a script to allow for cell analysis that doesn't involve humans counting things. So she's able to just highlight the section of the brain that we're looking at, and then have the computer actually count the number of neurons so that we know that we're not putting any human bias into it at all. And this is still relatively early, but data is holding true to what we saw with our antibody staining as well, that there's a decrease in the number of parvalbumin positive interneurons in the CA1 region of the hippocampus in the heterozygous females when compared to the males. Or sorry, and when compared to the wild type control. She's taken this one step further in those same mice and looked to see if there's any difference in the parvalbumin density based on the PCDH19 expression. So she's used a different mask to identify the stripe areas and then used her program to count the number of cells in each of those stripes. And at this point has not seen any difference in the cell density in the green areas, so the PCDH19 positive regions, when compared to the blue areas. So it may not be really related to the cells that they are directly attached to, or like the stripes that they're in, but there may be other differences at play. She's used the same program looking at the parvalbumin density in the cortex as well, and has not found any difference in the number of parvalbumin positive interneurons that in the cortex. And this is similar to what has been seen in a previous publication as well. So it means that the PV positive interneurons are making it to the cortex, but there might be something different about the hippocampus and hippocampus function that is changing the way that they're acting there. So that's kind of our big question right now, is why are there fewer parvalbumin positive cells in the CA1 of our PCE mice compared to the wild type controls? And to get to that question, there's a few things we need to look at. So could it be that there's not as many born? So are there not as many cells born in the mesoganglionic eminence? Or is it not as many migrate all the way to the hippocampus? So if they can't make it there, if their cell signals don't actually allow them to make it there, then it may decrease the number of cells who we see. Or is there more cell death? So once cells actually find their place into their final place in the brain, or the hippocampus, they undergo a process in which they really kind of refine their synapses, and some cells don't survive because you don't necessarily need them all. So are there more cells who actually, is there more cell death in the PCE mice compared to the wild type controls? So we started looking at this specifically in those embryonic mice, looking at the medial ganglionic eminence. And we do find some interesting things in that the cell proliferation is actually asymmetric in the cells that are PCDH19 positive or PCDH19 negative. So to do this, we have stained with a marker called Ki67, which is a marker for kind of newly born neurons. And you can look at them in the MGE. So these are the red cells here. And then we literally just counted the number of cells. So you can count all the blue dots there, and that's the blue dot is one cell. And then you can count how many blue dots also have green, and you can count how many blue dots also have red. And then you can take your proportion of those and figure out if there's more proliferating cells that are also green or PCDH19 positive versus the cells that are blue only, and those would be PCDH19 negative. And we did find some asymmetry in the number of proliferating cells that were PCDH19 negative compared to those that are PCDH19 positive. When you look at the wild type controls, there was no difference in the cells that were green versus blue and the number of proliferating cells. So this suggests that there may be some differences in timing and how the cells may be born, or that there's just not as many cells born in the PCDH19 negative regions, and that may affect their ability to migrate appropriately. And then we've done some other studies looking at if we can isolate the role of PCDH19 in the interneurons alone. And I'm so glad Dr. Umori already introduced this, because it's a little bit of a difficult concept. And again, he was very generous to donate these mice to us. So his conditional knockout mice, instead of crossing them with an EMX pre, we've crossed them with an NKX 2.1 pre. NKX 2.1 is a marker for early interneuron development. So the early born interneurons will express NKX 2.1, and when they do, the PCDH19 will be cut out of those neurons alone, and those neurons will express a green fluorescent protein, so that we can see where PCDH19 is knocked out, and it isolates them specifically to the interneurons. And so when we make these crosses, we end up with females who do not have the NKX, so they end up being wild type. We have females who are heterozygous in their interneurons. So half of their interneurons would have PCDH19 flocks out, and the other half would be wild type. And their males, since they're only expressing one allele, or one X allele, the PCDH19 would be knocked out on all of their interneurons. And then of course we have our wild type males as well. The first step we did with this is this fluorothel seizure latency model, in which other people have seen some differences in PCDH19 mice. It's a relatively easy assay to do, so we wanted to start with that one. And we have yet to reach significance, but there is a suggestion that our heterozygous females and then the floxed males have a decreased seizure latency, so a decreased seizure threshold when exposed to fluorothel. And then we can do histology on these and get these really beautiful pictures of, this is an inhibitory interneuron that expresses NKX 2.1. And with some very fancy microscopes, we can kind of assess where these are and look more at the synapses as well. And this is still very much pending work, but we're excited about it. We have a lot of further characterization to do with these NKX pre-floxed mice, including looking again at those hyperthermic seizure thresholds. And then I want to do some more EEG recording, because I am convinced that eventually we'll find some spontaneous seizures. In addition, there's a lot more histologic evaluation that we will be doing to look at the distribution of the synapses in the knockout neurons as well. So we still have a lot of work to do to try and figure out if interneurons are playing a role in PCDH19 pathophysiology. So a couple of things that we're looking at, we have a few other physiologic assays that we're looking for specific cell dysfunction, and specifically looking at those regions where the cells meet, so the green cells versus the not green cells, and looking if there's dysfunction between them. We're also looking at synapse formation, so we have a very cool marker where we can actually look specifically at the postsynaptic photons here and see if there's any difference in synapse formation in the cells that are mismatched. So the ones that are green synapsing onto the blue ones versus the blue ones synapsing onto the blue ones themselves, and then see if there's any changes in the number of synapses that are formed when it's specifically looking at the interneurons. And then we'd also like to look more at the exploration of this interneuron phenotype. Why do we see a decreased number of PP positive interneurons? And is it related to the proliferation like we talked about? Or is there actually a problem with migration and not enough cells are getting to the hippocampus? And then of course we have to look at cell survival and maybe more are dying. So, sorry, my talk's a little bit short, which is great. But in summary, the interneuron pathology is involved in many of the DEEs, and PCDH19-Clustering Epilepsy may actually be one of them as well. But there's a lot of other factors at play, and there's probably multiple roles of this gene. And I think it takes a village of us to try and figure out exactly what's going on and how we can best target it. But I would like to make a plug that further evaluation of this interneuron dysfunction can actually help lead to improved therapeutic targeting because there are so many DEs that really involve some abnormalities in interneurons. And with that, I have lots of thank yous. So Jack Parent is my mentor. He's helped me through all of this work. And as Julie mentioned, some of my first funding came from PCDH19 Alliance, and I'm really appreciative of that. It helped me to get to the point of NIH funding where I can hopefully continue to grow this work. And much thank you to the patients, because you guys really started it off. Thank you.
Test. If you could introduce yourself on the question that we all get to know each other, the first 8 minutes.
Hi everyone, I'm Sarah from Australia. I have a 9-year-old little girl with PCDH19, and I have 2 questions, if that's okay. Full disclosure, I only understand maybe every 6th sentence that you say. So forgive my ignorance with this question. But in regards to the cell segregation pattern and where they're located in the brain, you're saying that you know, obviously the brain of a mouse and the brain of a human are very different. Has it, I, for example, know of one individual who had brain surgery. I believe that piece of brain has been donated to Adelaide University. Is there any way for you to get your hands on that sample?
There's several, so Dr. Singhal can talk a little more about this too, but there's several patients who have had brain surgery with PCDH19, and that is something we have talked about, is getting our hands on it, and maybe we can talk as a scientific group about who's best to do some of those studies. But I think that would be really interesting to see the segregation pattern in the resected brain. Some of the individuals, I know there's an individual at UCSF, right, who had a brain surgery. It was a temporal lobectomy. So specifically taking out the hippocampus plus some of the neocortical temporal lobe as well. But yeah, I mean, I think that would be fascinating to see how much cell segregation is in those brain tissues, because we know that's where most of their seizures were coming from, right? Otherwise they wouldn't have had brain surgery. It's a great question, and we can talk more about that.
I'll courier over to you. And my second question was that, you know, we've heard from 2 researchers now, both who are focused on the seizures in their mice. Are there any studies done into other comorbidities that the mice may exhibit? For example, insomnia that a lot of our kids struggle with, or mobility issues, or in my daughter's case, I'm sure she's not the only one, autonomic dysfunction. Is there any way we can?
Yeah, so looking at those things specifically, we haven't looked at the sleep dysfunction, but it's relatively easy with some of our EEG data. So I think that's a great idea that we should look at it. Part of it is trying to have a robust phenotype. So trying to get something from a mouse that we can translate to humans. Some of the more subtle behavioral things can be really hard to translate into mice. So that's why there's a lot of focus on seizures, because you can obviously see a seizure, right? But you're absolutely right that there are so many other comorbidities that come with all of the DEEs that it's really hard to get to the bottom of in mice, because they can't do as sophisticated testing to be able to tell us. I don't, Sasha, have you done any motor testing in your mice. Basically normal, yeah. So I will tell you this is very, very preliminary, but we are hoping to use some AI technology to help us looking at some other motor changes in our mice as well. I have no idea if it's going to show anything or if it's even going to work, but the capability is there, and there's a lot of other D models that are using AI to kind of track the mice and see if there's subtle differences in their movements, their grooming behavior, their feeding behaviors, and things like that may be a little bit more translatable. So I think as our technology gets better, some of those things may come out from our mouse models as well.
Fantastic. Thank you. Thank you for all you do.
Thank you.
Thank you for the talk. I'm Brenda. I'm from Minneapolis, and I have a 14-year-old with PCDH19. And I'm very interested in the migration, and I'm wondering if you know or if there's literature that I may have missed on the intracellular domain and how that might signal, work to signal migration or changes in migration.
Oh, that is a tough question. I don't specifically know. Paul, do you know more about that? It's a really good question, though.
Yeah, there's some evidence that the intracellular domain can be cleaved off, so removed from the rest of the protein under certain stimuli. It's not actually very clear what that stimulus is, and can move to the nucleus and change the gene expression. So this is working in a way that we would predict a cell adhesion molecule to normally work. But how that connects with a migration phenotype is what I'm investigating.
And then Julie, back to the hippocampal. When you look at the animals and the loss of the inhibitory interneurons and the more prominent striping, have you noticed prominent asymmetries? And just getting back to these surgical patients that we have, I mean, it would stand to reason, right, that the randomness of all of this.
Yeah, and there are some where the hippocampus is all green, and then there's some that have on one side, and then they're very striped on the other side. So there's pretty significant asymmetries in some of the mice. That's where we're trying to get to the bottom of that, and I'm still struggling with the best way to figure out how to analyze that and relate it to the behavioral phenotype or the seizure phenotype that we see. So we started with this like stripe correlation, but I think we might have to refine that a little bit more as well. But I'm still convinced that there's something to the number of stripes, the number of abnormal cell interactions, and the location of those abnormal interactions as well. And I think that would play really nicely into the human phenotype, where some patients really have no seizures and they have maybe no phenotype, but they have some random exon activation because they're female.
That's just interesting, right? Because as you think about, there's very few patients that we consider for surgery because all the seizures seem to be coming from the left, or it still makes us nervous because we know that it's a genetic condition, and are we going to unmask something and we remove somebody's temporal lobe, which is not a benign thing. So, but it's nice to have the basic science to support that. In fact,
Maybe there's
an asymmetry, and it lines up.
And I think that's challenging for the humans too, because they're not all temporal lobe seizures, right? Some patients have very more classic frontal lobe seizures, though there is a large proportion that have what are more classically considered temporal lobe seizures. So it may just be kind of a location that is more likely to cause epilepsy and cause the clusters.
Going back to Sarah's comment earlier, I'm from the University of Adelaide, actually. So Joseph Getz, who's here, who's been very instrumental in this field, we collaborate with, he might be the guy who has the sample. I'm not sure. We maybe talk about it later. And if so, we discussed this possibility a number of years ago. The kind of difficulty is that, unlike the animal models where we have a control, here we've only got the test sample. So that only gives us nothing to compare to what might be considered what would happen in a brain that doesn't have polygastric epilepsy. And that makes the experiment really difficult to interpret. So I guess I could throw this maybe back at you, Joe. What prospect would there be maybe for a resection of other epilepsies? Could we get some kind of control to compare these precious polygastric samples to?
Yeah, I mean, we have lots of surgical samples, so, and we have lots of surgical samples from multiple different etiologies. So even, there's FDC5 surgical samples, there's, and then a lot of mesial temporal sclerosis that are not genetic, or what we presume are not genetic, but they, obviously everything's genetic in them.
Yeah. Can you just tell us a little bit about NKX3, from when Cree is expressed, which cell is expressed?
So my understanding of it is that it's expressed in the inhibitory interneurons. So the precursors for PV plus somatostatin. I don't remember some of the other ones. And it is expressed early. I think it's expressed as early as E8 or E9. So around the same time that PCDH19 is expressed.
Only in interneurons?
In the
At
interneurons.
E8?
The NKX?
It could be, yeah, NKX.
Yeah, NKX is relatively early. And forgive me, I haven't looked this up in a while, but it was around the same time that PCDH19 is expressed. I'll have to double check that. I could be wrong.
No, just wondering what may be causing the phenotype, of course.
Yeah.
Questions for Julie? And just to wrap up the surgery conversation, just so parents and families aren't running to the neurologist to say what you're when they had surgery. I mean, often a lot of our kids, when we do their EEGs early on, they have bitemporal seizures, right? And it's only for those very uncommon situations where whenever we hook them up to EEG, that they always seem to be coming from one side, that we then start to entertain surgery as an option. But surgery is obviously a big deal and can work for a select few, but it's probably not a good option for most. All right, thanks, Julie.
Thank you.